Welcome back to the Casual Ethnographer. In today's episode, we will be diving into part 2 in our series about evolution. This is the basics of genetics, the science that explains how traits are passed down, how life evolves, and ultimately how we are all connected. Now, genetics can get really complex, but don't worry, this is a crash course. By the end, you will understand the big ideas that help shape modern biology. Let's get started. Let's start in the 1800s with a monk named Gregor Mendel. He lived in what is now the Czech Republic, and he had a knack for science, but he also had some serious test anxiety. So instead of academia, he worked in a monastery garden, and that is where he made history.
Between 1856 and 1863, Mendel carefully bred pea plants, tracking traits like color, yellow or green, and texture, smooth or wrinkled. What he discovered was revolutionary. Traits don't blend randomly. Instead, they follow patterns.
Mendel figured out that each trait is controlled by two particles, one from mom and one from dad. Later, we gave those particles a name: genes.
Mendel's work gave us two big ideas. First, traits are determined by pairs of genes, one inherited from each parent. Second, when eggs and sperm form, each gene has an equal chance of being passed on. Today, we call that independent assortment.
Unfortunately, Mendel's 1866 paper was ignored for decades. It contradicted the leading botanists of his time. Only around 1900, after other scientists repeated his experiments, did his genius get recognized.
By 1900, science had also shown that all living things are made of cells. We knew that organisms grow because cells divide. In normal cell division, called mitosis, a cell makes an exact copy of its chromosomes. Humans have 23 pairs of chromosomes, so after mitosis, the new cells still have 23 pairs.
But when it comes to reproduction, things work differently. Through a process called meiosis, egg and sperm are made with just one copy of each chromosome. That's why we call them haploid. When egg and sperm join together, they form a diploid zygote, a brand new organism.
Genes come in different versions, called alleles. If you have two of the same, you're homozygous. If they're different, you're heterozygous. The combination of alleles is your genotype. How those genes show up physically, your hair color, height, or even pea texture, is your phenotype. Here's a real-world example. There is a condition called hereditary hemochromatosis, where the body absorbs too much iron. It's caused by a mutation in a single gene. I'm homozygous for this mutation, meaning I have two copies. My husband is heterozygous, one copy. Together we have one child who is heterozygous and another who is homozygous. Same family, same gene, very different outcomes. That's how genotype translates directly into health and traits. Now, a heterozygous pea plant might carry one allele for smooth peas and one for wrinkled, but you'll only see smooth peas in the phenotype because the smooth allele is dominant. The wrinkled allele is still there, hidden in the genotype, and can be passed to the next generation. That's a key difference between what you carry and what you show.
This is where terms like dominant and recessive come in. A dominant allele shows up in the phenotype, while the recessive one stays hidden until you've got two copies.
One common misconception is that dominant means more common or even better. That's not true. For example, having 6 fingers is a dominant trait, but it is rare. Dominant just means it shows up in the phenotype if even one copy is present. Another misconception is that every trait is strictly dominant or recessive. Reality is way more complex. Some traits blend, like flower colors, and others involve multiple genes working together, like human height or skin tone. In 1953, James Watson and Francis Crick, building on the X-ray work of Rosalind Franklin and Maurice Wilkins, described the structure of DNA, the famous double helix. DNA is a code written with 4 chemical bases, A, T, C, and G. A always pairs with T, and C always pairs with G.
That simple pairing rule is the foundation of life's diversity. DNA stores instructions for making proteins, which do almost everything inside a cell, from building structures, to carrying signals, to controlling chemical reactions.
Proteins are made from 20 amino acids, strung together in precise sequences. Some genes code for proteins, others regulate when proteins are made, and some don't code at all, but still play roles in how cells function. What used to be dismissed as junk DNA is slowly proving to be important. Genetics isn't just about understanding traits or diseases. It's revolutionizing our understanding of evolution itself. By comparing DNA sequences across species, scientists can trace how closely related different organisms are. For example, humans and chimpanzees share about 96% of their genome. That similarity isn't a coincidence, it's evidence of a shared ancestor. Genetics also shows us that evolution doesn't just happen through big, visible changes. Tiny mutations in DNA accumulate over generations. Some have no effect, others can be harmful, and a few give advantages that help a species survive. This is the raw material natural selection works on. Molecular clocks, tools that use DNA mutation rates as a timeline, allow scientists to estimate when species diverged from common ancestors. That's how we know, for instance, when our human lineage split from Neanderthals, or when mammals diverged from their reptilian relatives in the amniote lineage. And the field keeps evolving. Today, whole genome sequencing lets us track how bacteria develop antibiotic resistance, or how viruses like COVID-19 mutate and spread. It's evolution happening in real time, visible through the lens of genetics.
So while we've only skimmed the surface, you can see why genetics is central to biology and to our understanding of evolution. In the third and final part in the series, we'll take a look at how natural selection changes the frequency of genes in a population. The story of life is written in DNA, and it is still unfolding. That wraps up today's episode. As always, thank you for being here. I appreciate your presence. I'll see you next time.